Greetings, Bentley alumni, family, and friends. Again, thank you for joining us for today's virtual event. This is our second since we've all been working remotely. And this is the first week that our faculty members, like Zoe, are teaching remotely. So we're so appreciative of their participation. If you missed last week's talk from Rob DeLeo, please go ahead and check it out on our events page. We saved it on the Bentley alumni and family events page. So check it out. Today's featured speaker is Bentley University Lecturer Zoë Wagner. Zoe is a pharmaceutical scientist and bioengineer with a background in development of novel protein-based nanoparticle therapeutics for targeted treatment of solid tumors. Her research broadly seeks to understand how underlying biological structure and function can be used in the development of novel pharmaceutical interventions. So without further ado, please join me in virtually welcoming Zoë Wagner. Thank you, Zoe.
Thank you, Emily. Hello, everyone. Thanks again for joining. I am going to begin actually with just a couple of disclaimers before I get into any actual content. The first is that I am not a medical doctor, and nothing that I am saying right now should be taken as any sort of medical advice. If you have any questions about symptoms or your ability to receive a test, you should call your primary care provider, but again, not a medical doctor. And secondly, this is a rapidly evolving situation that we find ourselves in right now, and we are arguably just at the beginning of what is going to be a rapid acceleration in this country. And so information is changing on an hourly basis. So I am going to talk about what we know now. And that information might change. That information might change later today. It might change in a few days. It might change in a couple of weeks. But the only thing that we can do is understand the situation right now. We're going to have a lot more information available to us when it's all over. But because of that, I'm going to be sticking a lot to talking about the biology of the coronavirus and some of the underlying things that we do know. And I'm going to stay away a little bit from some of the more clinical information that, again, is coming in rapidly. So what is this? What are we currently experiencing, right? Let's start with the basics. Let's do some background. So the pandemic that we are currently experiencing is caused by a virus that is called SARS coronavirus 2. And SARS stands for severe acute respiratory syndrome. It's named after the condition that it causes. And it is called SARS-CoV-2 to differentiate itself from SARS-CoV-1, which caused the SARS outbreak in around 2001, 2002. So the virus itself causes a disease called coronavirus 19 or COVID-19. So you will see all of these terms being used pretty much interchangeably in news media. You can assume more or less that if anybody says coronavirus, they are talking about the current SARS-CoV-2. Technically, if you're talking about the disease, then you're talking about COVID-19, but again, everything is being used interchangeably right now. So the coronavirus gets its name from what it actually looks like. If you take the virus and you put it under a microscope, what you will see is a spherical viral particle that is completely surrounded by this halo or corona of spiky proteins. And so because it looks like it has this halo or corona around it, we call it a coronavirus. And coronavirus 2 is one of 7 coronaviruses that are known to infect humans. 3 of them are known to cause severe disease, while 4 of them cause very moderate and mild symptoms. So since we're talking about the coronavirus, it probably behooves us to begin with the question of, what is a virus? What are we talking about here? So a virus is a small infectious agent that can only replicate inside a host. And you can have an equally scientific and philosophical argument over whether a virus is alive or whether it is not alive, but a virus requires a host. And this makes it very different from other organisms that we think of causing disease, such as bacteria. So if you were to take bacteria, such as E. coli, for example, and you were to take it into a lab and put it in a plate and just sort of leave it, it would begin to grow and divide and multiply. The same thing would not happen with a virus. The virus on its own is incapable of replicating. It requires a living host cell. And that living host can be an animal, it can be a plant, it can be a bacteria. It requires a living cell to replicate inside of. So how do viruses make us sick, right? What are they actually doing inside the body? So when we are exposed to a virus, and this could be because, for example, a person who is infected has coughed and expelled viral particles onto a surface, and then later a healthy person comes by and touches that surface, and then maybe they touch their mouth or their eyes, somewhere on their face. The virus now has received entry into that healthy person. And the virus, in the case of coronavirus, is going to make its way specifically to the lungs. And once it reaches healthy cells in the lungs, it is going to use those spiky proteins that are organized along the outside of the virus itself to attach to the healthy cells. And once it's attached, it's actually going to enter into the healthy cell, and it's going to force it to start making more viruses, right? So it's actually going to sort of hijack the healthy cell itself and turn it into this production factory for making new viral particles. And as the cell begins to make more and more of these viruses, they are going to be released, and those viruses can then spread within the body and continue to infect more cells. So this infection of healthy cells is what caused the symptoms of the disease. So coronavirus, as we know, it's called SARS coronavirus 2, severe acute respiratory syndrome. So we know that it is predominantly affecting the lungs. The way that it's doing this is that in a healthy person, the lungs are the location where oxygen exchange happens, right? We breathe in oxygen from the air moves into our lungs and down into tiny sacs called alveoli. If we all maybe try to remember back to arguably high school biology, and you might remember an analogy of lungs looking sort of like trees, right? That you start with the central branch and then you move into smaller and smaller branches and you end with almost leaves. And in this analogy, the alveoli are these tiny little air sacs that are at the very end of our lungs, and that is where oxygen exchange is actually happening. So these tiny little air sacs are covered with a web of very fine blood vessels. And the oxygen from the air is going to move from the inside of the alveoli into the blood vessel and then provide oxygen to our entire body, which is what we need to carry out all normal physiological functions. So in a healthy person, that is how oxygen exchange happens, right? It happens in these very important alveoli surrounded by this network of blood vessels. Now, when the coronavirus infects lung tissue, what happens is that it causes damage both to the cells of the alveoli as well as the cells of the blood vessels. So when the virus takes over normally healthy cells and turns them into these viral production factories, eventually the cell can't handle it anymore, and the cell itself will die. And as more cells become infected, more cells begin to die. And this results in an accumulation of really just debris, leftover pieces of cell that have unfortunately died because of this viral infection. And as more cells of the alveoli and the blood vessels begin to die, what happens is that fluid begins to leak into the lungs. Normally, we have a very tight barrier in the lungs, and that's because we need there to be a clean space for air and oxygen exchange to occur. As fluid begins to leach into the alveoli and into those air sacs, it becomes much more difficult to breathe. It becomes much more difficult for oxygen exchange to occur. And this is a condition that many of us have probably heard of. We refer to it as pneumonia, right? Fluid accumulation in the lungs. And many people who have moderate to severe infections with the coronavirus are initially presenting with pneumonia, with this buildup of fluid in their lungs, which again is caused by the virus damaging the cells that are lining the alveoli and lining the lungs. So the pneumonia and this damage to the lungs, in some people, the body is able to recover from that, right? And your immune system will kick in. It will recognize the virus is not meant to be there. It will begin to attack the virus and it will try to clear it from our system, right? However, in some people, this becomes even more severe and it advances beyond pneumonia into a condition that is called acute respiratory distress. And acute respiratory distress is further degradation of lung function, where the damage to the lungs continues to become even more severe in patients. And the treatment that we have for acute respiratory distress, if it reaches a certain level, is generally putting people on ventilators. And ventilators are machines that are going to mechanically move pressurized air into and out of the lungs and sort of take over the role of our own muscles in enabling oxygen exchange. So these ventilators can be very helpful. Unfortunately, they are not always helpful. There is still a biological limit that our lungs are able to withstand. And unfortunately, some people do have complete respiratory failure and the ventilator is not able to help them. It is also worth noting that this is a significant limitation for our healthcare system right now. We have, by some estimates, 160,000 ventilators in this country, total. That is the availability of everyone in the United States. And so if too many people become infected and have a severe respiratory distress, develop acute respiratory distress, we may reach a situation where the number of ventilators is underserving the number of patients. So some number of patients, and we don't have good numbers on it, so I am not going to try to give you a percentage for the number of patients who develop acute respiratory distress, but some number of patients do develop this very severe illness. Interestingly, it seems like there may actually be a second factor that is coming into play with people who become severely ill due to the coronavirus. And that goes back to something that I said earlier about the immune system. So the function of our immune system is to recognize foreign invaders and protect our body from them, right? To recognize when something that is not supposed to be there, to be in our bodies, is present, and to get rid of it. And normally, that is a very good thing, right? Our immune system is meant to recognize the coronavirus as foreign. It is meant to attack it, get rid of it, so that we can become better. However, in some cases, it appears that the body's response to the virus is too severe, that the body over-responds, and that there is an overreaction of the immune system that leads to too strong an immune response. And this causes something that is called a cytokine storm. And in a cytokine storm, this overreaction of the immune system leads to a flood of immune cells into the lungs. And this can actually end up doing more harm than good in some cases. So for example, if you have a sort of normal level of immune response, one thing that your immune system might do is begin to open up blood vessels in the lungs a little bit to allow cells of the immune system to enter into the lungs and better help combat the virus. And that would be a good response from the immune system. On the other hand, if the immune system responds too severely, it might open up those vessels too much, and that would allow even more fluid to come into the lungs. So there's some indication that, again, in some number of patients, we are seeing this response where it's actually the immune system over-responding that is causing a problem with the patients, where their own body is reacting too severely to the coronavirus. So one of the more interesting things that we have noticed about the coronavirus is that there is a very broad range in terms of how people are presenting clinically with symptoms of the disease. Looking at data coming out of China, and I think that for now that is probably one of the better places for us to get our information, because China is in the recovery phase from their experience with coronavirus, whereas the United States is still very much in the acceleration phase of our experience with this virus. So China has a better ability to provide somewhat complete information, whereas the United States, again, does not to the same extent for right now. But so in China, about 81% of the people who were confirmed to have coronavirus presented with very mild symptoms. They had a fever, maybe they had a cough, but they were pretty much okay. An additional 14% of patients presented with severe disease, and this included shortness of breath and low oxygen levels in their blood. So this is, again, going back to that fluid accumulation in the lungs, making it very difficult for oxygen exchange to occur appropriately. And then lastly, 5% of the cases were classified as critical. And so those were patients who had respiratory failure or who further developed multi-system organ failure. So there is that range in that, again, about 81% of people had very moderate symptoms, but some people had very severe symptoms. So one of the things we've been trying to understand about the virus is, why do people respond differently, right? Why are some people presenting completely asymptomatic, mild symptoms, or incredibly severe disease? And one of the factors that we think is definitely coming, is most likely, I should say, I'm going to try to qualify everything that I'm saying in case it changes, but one of the most significant factors for now seems to be age. And we can look at how age affects your likelihood of dying from the disease or having a severe case of the disease. And the coronavirus actually has a very distinct effect according to age. What we're used to seeing in many viral diseases is almost a U-shaped curve, where the people who are most affected, most likely to have severe disease, are the very young and the very old. And these are people who, A, have very poorly developed immune systems, or people who, because of their advanced age, have weaker immune systems. And what we see with the coronavirus is actually not that distribution of severe disease in young and old. We are actually, for now, only seeing severe disease in older people. In fact, looking at younger populations, in children between the ages of 0 and 9, if we look at the cases in China, there was actually a 0% case fatality rate amongst children ages 0 to 9. And that appears to be fairly consistent in that most young children are presenting with very mild disease if they have symptoms. That case fatality rate increases to about 0.2%, again, looking at numbers from China, in people who are between the ages of 10 and 39. And then the highest fatality rate is seen in people over the age of 80, where it reaches about 15%. So age definitely seems to be a factor in the likelihood of developing severe disease. Older people are more likely to have a weaker immune system, and they're also more likely to have pre-existing conditions. Pre-existing conditions have also been looked at as being linked with a more severe disease outcome. So people who already have diseases such as heart disease, who already have diabetes, respiratory disease, high blood pressure, all of those things appear for now to be likely to increase your likelihood of having a more severe response to the coronavirus. So when we are looking at the coronavirus and the implications of it and what it is, what it might do in our population, our global and national population, one of the things that we might want to do to understand the severity of it is sort of look at it in comparison to previous coronaviruses. And there are a couple different metrics that we use in order to talk about this in a comparative sense. And one of those is the case fatality rate, which I just mentioned. And the case fatality rate is the total number of deaths from disease divided by the total number of cases, right? So how many people have confirmed to be tested positive? And of those people who have confirmed to test positive for COVID-19, how many have passed away because, as a result of the disease? And this is a number that you're probably going to see reported a lot in the news. And it's something that people are talking about a lot. I do think that there is a very important point to make about this number. in that it is completely dependent on the number of confirmed cases of disease, right? That is the denominator in this number. And the number of confirmed cases of disease is itself going to be dependent on what population are you testing, right? And are there qualifiers for the population that you are testing? So if you are only testing a population of people who are already presenting with moderate to severe disease, you, to begin with, have a fairly small population that you are testing, and they are already quite ill, right? And so because they are already quite ill, they may be more likely to unfortunately not recover from the virus to begin with. So the case fatality rate in that case can be artificially skewed such that it appears that the disease is much more fatal than it actually is. If you have expansive testing where many, many individuals are tested, regardless of how severe their symptoms are when they present, then your case fatality rate may appear lower. And this is something that we can see reflected in China and their initial numbers when they were looking at the case fatality rate due to COVID-19. Their initial estimates when they were just beginning to deal with the disease and the originating outbreak in Wuhan was that the case fatality rate due to COVID-19 was somewhere between 17 and 20%. Over time, as they were able to implement more testing and were able to expand the denominator, the number of total confirmed cases of disease, that number ultimately fell to about 3.9% as their case fatality rate. So the case fatality rate is subject to change. It is completely dependent on how many people have been confirmed positive. And the number of, again, those confirmed positive cases is going to be who are you testing, right? So the case fatality rate can change over time. It can also change when you look at different populations, like those different age groups that I just mentioned. Right now, the COVID-19 appears to have a global case fatality rate of about 3.7%. And that is globally. So that is a global average. There could be higher case fatality rates in some countries and lower in other countries. Again, depending on how broad the testing and case percentage has been within different countries. In the United States, we could have very different case fatality rates in individual states and then sort of a national average. And we can compare this to previous coronaviruses. So the two previous coronaviruses are the SARS outbreak that occurred in 2002, and then MERS, which is the Middle Eastern Respiratory Syndrome, in 2012. And SARS had an estimated case fatality rate of about 10%, whereas MERS had an estimated case fatality rate of 34%. So in comparison to previous coronaviruses, COVID-19 is actually appearing to be significantly less fatal. And this is, on the one hand, a good thing, right? That it is less likely that people who are positive for the disease may die from it. However, it is worth noting that both SARS and MERS infected far fewer people. SARS only infected about 8,000 people, whereas MERS infected 2,500. And we could also compare this to the seasonal flu, which has a case fatality rate of about 0.1%, according to the CDC. So the COVID-19 is significantly more dangerous than the seasonal flu. So case fatality rate is one metric that has been discussed a lot. There is frankly so much we could say about case fatality rate and what it takes into account and what it doesn't take into account, and other numbers that might be better, other numbers that we will hopefully have in the future. That frankly in and of itself could be its own point of discussion, but because it is so widely publicized, it is something worth discussing. And again, that number is almost certainly going to change, right? Again, we are talking about the numbers as they appear now. The numbers are unfortunately different depending on if you were getting your information from the CDC, the WHO, the European CDC. But again, we're working with the information we have now. So another metric of comparison that we can look at is something that looks at how easily transmissible is the disease, right? How easily does it spread within a population? And we can measure that with a value called R naught. And R naught is the average number of people who will catch the disease from one infectious person. So if I am an infectious person, how many people am I likely to infect? And the R naught value for previous coronaviruses for SARS, it was about 3, range somewhere between 2 to 4. For MERS, it was somewhere between 2.5 and 7. And for the COVID-19, again, this is still an evolving number, but for now, it seems that most estimates put it somewhere between 2 and 2.5. And this means that the COVID-19 is less likely to be spread than SARS or MERS, the previous coronaviruses, but it is more likely to spread than the seasonal flu, which appears to have an R naught of about 1.3. Now, one of the things about this R naught value that's very important to note is that it can be controlled, right? In that we can change the transmission rate of the disease by decreasing the likelihood that an infected person is likely to come into contact with non-infected people, right? And so this is why we have implemented really strict social distancing and really tried to impress upon people the importance of isolating themselves physically from other people. And this is to prevent the transmission of the disease, right? Prevent the number of healthy people that one infected person can infect. And this plays into something that you've probably seen a lot in the media, which is the idea of flatten the curve. And so when we talk about that, what that is talking about is the total number of infected people over time, right? How many people are infected with the coronavirus over time? And the reason that it's important to think about that is because our healthcare system has a maximum carrying capacity, a maximum number of patients that we are able to deal with at any given time. And that is dependent on multiple things. It is dependent upon how many beds are there in the hospitals. Right now, as of best estimates from 2011, we have about 3 hospital beds per 1,000 people in the United States, right? So that is going to potentially be a limiting factor. The carrying capacity is also going to be dependent on how many healthy staff members do we have in hospitals, how much protective equipment do we have available for those healthcare workers who are in hospital settings, and simply how many resources do we have in hospitals. So we have a maximum carrying capacity in our healthcare system. And if we have incredibly rapid spread of disease and a lot of people become infected very, very quickly, what's going to happen is that we are going to very rapidly overwhelm our healthcare system. And the number of people who need healthcare support, who need medical assistance, are not going to be able to receive it because we will be completely overwhelmed, right? There will not be enough capacity for the number of people who are ill. If, on the other hand, we can slow transmission of the disease and make it so that the number of people infected sort of plateaus over a longer period of time, then we might be able to maintain the number of people who are ill and require medical attention at a level that is better able to be supported by our healthcare system. So this plays into something that I'm going to talk about just because a lot of questions were asked about it probably towards the end, and questions about how long might this whole thing last, right? And when we're looking at the capacity of our healthcare system, in many ways, it's actually better if it lasts longer. We do not want to overwhelm the healthcare system by having incredibly rapid spread of disease very, very quickly, to have a huge spike and then everything, and then a decrease in the number of patients. But so this, again, everybody's role for right now is keeping apart, right? Physical distancing, ensuring that you are coming into contact with as few people as possible to decrease the transmission of the disease. So for now, we know that COVID-19 is here. We know it's a significant strain on our healthcare system that is probably only going to become more significant in the coming weeks and months. So what do we do, right? There are two different approaches that we are currently working on. One is a short-term plan that relies on trying to find drugs that can be used as quickly as possible to begin to treat patients with and try to decrease the infection with coronavirus and improve symptoms. And then the longer term plan relies on developing a vaccine to ensure that we can build immunity in the population and prevent infection. So talking about drugs and potential treatment options first, the one that I'm going to start with is one that has been in the news a lot within the past few days, and that is a combination of two drugs, the first one being hydroxychloroquine and the second being azithromycin. So hydroxychloroquine is a modification of a drug called chloroquine, which has been used for decades in the treatment and prevention of malaria. And azithromycin, on the other hand, is an antibiotic. It kills bacterial cells. So we have some data in lab-based tests to suggest that hydroxychloroquine is effective in preventing infection with the SARS coronavirus 2. And most of the sort of public interest in this drug combination, the hydroxychloroquine and azithromycin, appears to come from a French study that was performed and published a few days ago that looked at the efficacy of treating patients that are confirmed to have coronavirus with this drug combination. And so what this study did is that they had 36 patients, and they looked at treating patients with hydroxychloroquine alone, with a combination of hydroxychloroquine and azithromycin, and with standard of care, so no specific drug treatment. And what that study found was that after 6 days, the patients who received standard of care, so no specific drug treatment, about 12% of them were able to recover from the infection, were able to recover from COVID-19. Of patients who received hydroxychloroquine alone, 57% of them were able to clear the infection. And of patients who were treated with hydroxychloroquine and azithromycin, 100% of them were able to clear the infection after 6 days. Now, that appears very promising on the surface. However, it is very preliminary, and I would urge extreme caution before anybody takes anything scientifically sound from this study for several reasons. The first one being, there was a very small patient sample. We only looked at 36 patients, and only 6 of those patients were treated with this combination of hydroxychloroquine and azithromycin. So only 6 patients. 6 is what is the number that we are looking at here. Further, those 6 patients tested negative for the disease after 6 days of treatment. However, 2 days later, so at day 8, one of those patients tested positive again. So it seems like there is certainly some evidence to suggest that the patients were not actually cured. Rather, the viral infection level decreased below the limit of detection and then came back when the therapy was stopped. So that is a concern. The size of the patient sample is a concern. The study was performed on patients who were exhibiting a very wide range of symptoms, from some who were completely asymptomatic, some who were mild, some who were more severe, and we don't know how those patients were put into individual treatment groups. And so there's a lot that we don't know about this study. So before we can take any good clinical or scientific information from the study, we need to repeat it in a more properly controlled fashion and with much larger numbers of patients. So while it is certainly promising, I think that a significant amount of moderation in the way that we're speaking about it is warranted, because there's frankly just not enough information yet to suggest that it's going to be an effective combination of drugs. One of the other drugs that is being tested is a drug called remdesivir. Remdesivir is a drug that was initially developed by the pharmaceutical company Gilead, actually to treat Ebola, and is now being used to try to treat COVID-19. We know that it works in the lab. We know that it works in some animal models, and clinical trials are ongoing. There is one trial that is finishing up in China, and we expect that to be reporting initial results in early April. So within the next few weeks, hopefully, we will be receiving the first results of the trial looking at the efficacy of remdesivir in patients who are positive for COVID-19. There are also 3 trials currently ongoing in the United States looking at the efficacy of this drug. They have not reported yet, but we are looking at the efficacy. And the WHO, the World Health Organization, has also recently initiated a large global trial called Solidarity. And the aim of that is to test what they think of as the 4 most promising drugs in the treatment of COVID-19. They are testing remdesivir, they're testing hydroxychloroquine and chloroquine, and then they are testing 2 drugs that are currently used to treat HIV, lopinavir and ritonavir. And so this trial is going to be again, global, hopefully enrolling thousands of patients, and will hopefully get off the ground as quickly as possible to test which of those drugs, if any of them, are going to be most effective in treating COVID-19. Now, it's really important to note here that these trials are moving forward, but the timeline is, again, unfortunately unclear. One of the things that we need to ensure is that the drugs are not just effective, but they are safe, right? We cannot sacrifice safety for the sake of speed when we're trying to find a treatment for COVID-19. We need to ensure that we are still performing all of the necessary safety tests to make sure that we are not going to make people who are already ill more sick in treating them. So depending on the results of these trials, as they begin to report, we may begin to see formal recommendations put in place for what the drug standard of care treatment should be. But until we begin to see reporting from some of these clinical trials, we won't really know the answer to any of these questions about what is the most promising drug candidate moving forward. So there are a lot of drugs that are currently being tested. Hopefully some of them begin to show positive results. And the other direction that people are going in is attempting to develop a vaccine. So the idea with a vaccine is that you train your body to recognize what the virus looks like, so that if you are exposed, your immune system can immediately say, that is bad, I'm going to attack it. And you will not have as severe a presentation of illness. So in order to do that, though, we need to develop a vaccine that will effectively train the immune system for how to respond should you be exposed to the SARS coronavirus 2. We have initiated a clinical trial in the United States. The Massachusetts-based company Moderna initiated a test of their mRNA vaccine that started just last week. I think the first dose, I believe, was administered on March 16th. And that trial is going to be performed in 45 healthy patients. Those patients are going to receive 2 injections of the vaccine 28 days apart. We are going to be looking at 3 different doses of the vaccine to look at mainly just safety. So all of the people enrolled in this study are going to receive 2 injections of the vaccine, and they are then going to be monitored for a year afterwards for any side effects that may occur. So in order to ensure that any vaccine that is being developed is both safe and effective, it will go through several phases of testing. This phase is the first phase, and the phase 1 test is looking just at safety. It's looking at, are there any side effects that arise because of injection with the vaccine? So that could include things like muscle soreness. It could include injection site pain or redness or swelling at the site of the injection site. We're going to look to ensure that the vaccine is in healthy people safe. If that is successful, we will move into phase 2, which is looking at hopefully several hundred volunteers, looking still at the short-term side effects, but also looking at how the immune system responds. Is the immune system effectively being trained to identify the coronavirus and clear it in the future? Are we seeing the immune response that we would expect? And then lastly, phase 3 studies would hopefully be initiated, which would then have hundreds, if not thousands of volunteers, which would be continuing to monitor safety, looking at side effects, and then looking at how people who get the vaccine compare to people who do not. So this process is lengthy. The fastest that this could happen would probably be on a timeline of 12 to 18 months. 18 months, I think, is a much more reasonable timeline for this. And again, that is because we cannot sacrifice safety for expediency. We need to make sure that even though we are trying to develop everything as quickly as possible to meet the current demand, that everything that we are administering to patients and to the general population remains safe. So the vaccine will probably not come for quite a while. It is entirely possible that the vaccine will be developed after there has been a peak and then a regression in the number of cases of the COVID-19. But it is currently in development. Again, Moderna is currently the furthest along in terms of its clinical trial. However, there are a large number of vaccines that are in preclinical development that will probably begin testing soon. So there will be multiple options for multiple vaccine candidates being brought forward at the same time. Should any of them prove to not be effective, there will hopefully be backups available. So in terms of looking at the basic biology, looking at some treatment availability, that is a very small summary of some of the information that we currently know. I think that just as a final note, there are a couple of things to remember. One of them is that our job right now, everyone, is to practice physical distancing. And I'm going to echo what my colleague Rob DeLeo said last week, in that I think this needs to be rebranded. We should all be focusing on physical separation, but not social distancing. We should be focusing on connecting socially with as many people as we can, virtually, online, not in person. But as this period of uncertainty and of physical distancing moves from weeks into months, I think it is going to be very important to maintain those social connections, which we know are so important for our mental health and well-being. So now would be a great time to reach out to friends, to colleagues, to family, to check in, to make sure that people are okay, to spend a little bit of time every day just not thinking or talking about the coronavirus. I think that would probably be very helpful for everybody's mental health. Now is a really good time to practice some kindness, to practice some generosity if you are able to practice really, really good hand washing hygiene, and to ideally, again, physically separate yourself from others so that we can give our healthcare system as much of a chance as possible. So I am happy to answer any questions that people may have that were either pre-selected or came in as we were going. Thank you.
Thanks, Zoe. Okay, we have lots of good questions here and only a few more minutes, about 15 more minutes to respond. So let's turn through some of these questions, shall we? Betsy Packale, and I apologize if I pronounce your name incorrectly, a graduate from the class of '93 asks, when will this virus crest and when can we get back to work? Which is a very popular question, of course.
Right. I think that's probably what most people are wondering right now, is just when is it all going to be over, right? When are things going to go back to the way they were before? And that's a really difficult question to answer for right now, unfortunately, because in the United States, we have very different regulations on a state-by-state level in terms of how severe restrictions are for this physical separation. So we can't quite say how fast the disease is going to spread within the United States population. I can say that there have been many attempts to model this, and there have been attempts to model it assuming very rapid transmission of disease as well as slower transmission of disease. So if we assume fairly fast transmission of disease, meaning that a lot of people get it very quickly and then the number of cases decreases, most of that probably puts the peak of this happening somewhere in late May. Again, maybe. So a colleague in National Applied Sciences, Tony Kashesky, recently did a model that showed about 3 months for the peak in the United States. again, with very high transmission rates. That is in many ways the worst case scenario. Yes, it would get us all back to work as quickly as possible. However, it would also put the biggest strain on our healthcare system. If the disease spreads more slowly, we give our healthcare system a better chance. That, in many of the models, will put the peak sometime in September, if not later, maybe sometime late summer. So when this all ends is going to be very, very much dependent on how much does it spread in the population, which is going to be how much opportunity do we give it to spread in the population.
Great. And one follow-up question to that, which might help people think about this, especially as we're all social distancing, is the length of time that this virus survives on materials and surfaces. We had several questions submitted about that. And what is the simplest way to clean paper, cardboard boxes, etc.?
Right. So there have been studies looking at the stability of the virus on surfaces. And so the interesting thing about viruses, again, is that, you know, they're not quite alive. However, they can persist and be infectious on surfaces, meaning that the virus itself remains intact. When the virus, quote unquote, dies, what that means in a surface level is that the virus would effectively sort of fall apart, that outer shell would disintegrate in some way, and the component pieces would sort of come out. So some studies have shown that coronavirus can persist for a pretty significant period of time. It seems that it's stable in aerosols for about 3 hours. So that means suspended in the air, suspended in air particles for about 3 hours. It appears to persist on cardboard for 24 hours, and then on less porous materials such as stainless steel and plastic for 2 to 3 days. So that is the length of time when the virus appears to still be virulent, capable of causing infection. So because of that, cleaning things makes a lot of sense, right? Ensuring that if you touch something that somebody else has touched, you want to wash your hands pretty quickly, that hopefully you want to wipe down surfaces that are commonly touched in your household. And per the recommendations of the CDC, there are several different solutions that you can use to inactivate or kill the virus. It seems that anything with an alcohol concentration between 62 and 71% is effective in killing the virus. So 62 to 71% ethanol should be good. In addition, you could make a 0.5% hydrogen peroxide solution, which would also kill the virus, or a 0.1% bleach solution. So just household bleach, what we sort of might use in the laundry, for example. And again, per the CDC, for the bleach solution, that's about 1/3 of a cup per gallon. Now, I just want to point out something very important about that, which is that in people's very supported efforts to clean their houses, they should not mix cleaning supplies. So you should not try to create some sort of super cleaning solution by mixing any of these things together. You should never mix bleach with pretty much anything else. So certainly mixing bleach with vinegar, ammonia, or rubbing alcohol will actually create incredibly toxic compounds that can be very, very dangerous. And in addition, mixing hydrogen peroxide and rubbing alcohol can also be very, very dangerous. So all 3 of those solutions that I mentioned can be used individually, but please, please, please do not combine them.
Great. And now pivoting to a popular question as well, this one submitted by Justin Crotty, MBA 94, wanting to know the similarity of this coronavirus with the seasonal flu, and will it sort of be a similar situation going forward? Are there vaccines? Somebody else asked, are there vaccines for the other 2 coronaviruses that we just haven't heard of?
So when we look at the availability of vaccines for previous diseases, most of the vaccines are going to be, they need to be specific for the virus causing disease. So a vaccine for example, influenza, or a vaccine for something like H1N1, the swine flu, would not be something that we could use for this coronavirus. And we need to make a vaccine from the ground up. Right? We need to build a new vaccine to be effective in treating in preventing, I should say, the SARS-CoV-2. So we have, in the past, unfortunately, developed vaccines a little bit behind the curve for when they would be most effective. And that's because, again, the timeline is simply, there's too many tests that need to be done to ensure safety. And we really cannot sacrifice safety for the sake of trying to get something out as faster. I think I remember one of the questions being having to do with the flu, asking, should you get a flu shot, right? Is there any reason to get a flu shot to protect you against this? And unfortunately, it will not protect you against the coronavirus. It will not provide any manner of immunity for you. However, one of the really important things that people having a flu shot will do is prevent hospitalization due to the flu. In this current pandemic situation, we've kind of forgotten about influenza a little bit, but I think it's important to note that there have been 29 million cases of influenza this flu season. There have been 16,000 deaths. There have been about 280,000 hospitalizations. If we can decrease the stress on our healthcare system due to the flu, that frees up space for dealing with patients who have coronavirus. So from that perspective, again, there's no direct comparison, but we can hopefully clear up space within the healthcare sphere. In terms of whether the coronavirus will behave like the seasonal flu, it's sort of hard to know for now. One of the things about the flu is that, as the name, it is seasonal, right? It cycles. We get it pretty much at the same time every year, give or take a few months. And we see it move in and out of the population. What we call the flu is actually usually a different virus or combination of viruses every time. We don't know yet whether coronavirus is going to hit us and leave, or whether it might cycle and be sort of a part of the normal viral sphere that we are exposed to. That is possible. That is one of the things that appears to some extent to have happened with swine flu, but we don't know yet if it's going to be something that happens with the coronavirus.
There are so many good questions. I wish that we had time to get to all of them, but we probably only have time for one more. This question, I know there's been a lot of rumors going around, and so an anonymous attendee asked about the use of ibuprofen, and is it dangerous to take if you have coronavirus, and why would that be?
So I have not seen anything suggesting that it would be dangerous to take ibuprofen if you have the coronavirus. Okay. And again, not a medical doctor.
Right.
Not a medical doctor, just going to make that as clear as possible. However, for now, because we don't have specific treatments available for the coronavirus, we can't tell people this is the drug combination that we are going to give you. Many people, again, are presenting with those sort of very mild symptoms, in that maybe they have a fever, maybe they have a cough. And so if they go to their primary care physician, that person may say, just sort of weather it. It's kind of like the flu, and hopefully you improve. Or they may say you can take some over-the-counter drugs to try to ease symptoms, such as maybe decrease your fever, such as maybe ease muscle pain. But I have not seen, I should say, any clinical suggestion in the research thus far that would suggest in a well-researched way, I should say, that there is any negative correlation between taking ibuprofen and the coronavirus. I haven't seen it in the literature for now.
It hasn't hit the scientific journals, it seems, so that's your area of expertise. So we'll keep waiting. As we know, things are changing every day. Maybe we have time for one more question. One of the questions is the recommendation of going outside if it lives in the air for 3 hours. People who want to continue to support restaurants who are doing takeout business, sort of what is your advice for that?
So the idea of the virus living in the air for 3 hours, so we know that it can stay within aerosols. So the question then becomes, how long are those particles going to be suspended in the air, right? How long are they going to stay there? And then how long is the virus, again, will probably remain viable for about 3 hours. So one of the main things is the particles are more likely to remain suspended in places that are closed and have no airflow, right? So not entering small spaces with other people would probably be advisable to prevent from possibly inhaling any sort of viral particle from the air. So that, again, physical distancing would sort of cover that. If you are outside, if you are out in the world, in nature, there's probably going to be breezes, there's probably going to be movement of air. I think that currently most recommendations, even of states that have shelter-in-place rules, are that people can still go outside. That if you are walking around outside, you should maintain still your physical distancing of 6 feet, but you should not be concerned about contracting it from the air. Most people, when they cough, they are able to expel particles about 6 feet, which is why we have created that benchmark, right? So at the farthest, they can expel probably about 6 feet worth of viral particles. In terms of continuing to support local takeout, on the one hand, yes, we should probably be doing everything we can to support local business right now. And that means that if at all possible, maybe getting takeout. Because the virus is able to persist on cardboard and on plastic for longer periods of time, if you would like to be as careful as possible, you could wipe down the outside of those containers, right? You could make yourself your bleach solution, your 0.1% bleach solution, and you could just wipe down the outside of all of your takeout containers, then wash your hands, because you also don't want to eat bleach, and then hopefully enjoy your meal. And, you know, in that case, most evidence seems to suggest that you should be fine.
Great. Thank you for your suggestions. It's very helpful as we all navigate this together. Thank you again for your time this afternoon. We really appreciate it. And we will be sending out the recording to all of our participants. So thank you for all of you who have joined this afternoon. We'll also send our schedule of upcoming virtual events, and we hope that you'll join us again soon.
All right, thank you.
Thank you.